thanks for the introduction, and thank you everyone for coming out today to listen to my defense. It is titled A Taphonomic Sedimentologic and Geochemical Analysis of Cetacean Bonebeds from the Middle Miocene from the Topanga Formation in the San Joaquin Hills, Oragne County, California. And first, I'd like to thank everyone who made this moment possible for me. Of course, Dr. Adam Woods, who has been my thesis advisor, who has been eager to give constant support throughout my thesis project, and of course Dr. Jerry Lips, who is the director of the Cooper Center and thesis committee member, who has given me access to the Bonita Canyon bonebeds and provided expertise and a place for me to conduct my research at the Cooper Center. Also, Dr. Nicole Benuso, who will be providing me with valuable feedback during my thesis review period, and all of the staff and professors here in the geology department and staff at the Cooper Center, specifically Meredith Riven, Gabriel Santos, Michelle Barboza, and Crystal Cortez, who showed me the ropes and assisted me while I conducted my research. And of course, my husband and mother. My mom couldn't make it today, but my husband's here, provided me much needed emotional support and babysitting services. So bonebeds are stratigraphically thin accumulations of vertebrate hard parts, which consist of multiple individuals from either one or multiple species. And cetaceans are aquatic mammals, namely whales, porpoises, and dolphins. And during this presentation, I'm referring to whales. So Brett and Baird, 1986, divided taphonomy into two general phases. First is biostratonomy, which are the mechanical, chemical, and biological processes that affect an organism from the moment it dies until its final burial, and can be further broken down into 4 sequential sequences or stages. First is transport and reorientation that occurs to the carcass once the organism dies, and typically immediately follows death. Then it's followed by disarticulation of the skeleton, which occurs and is followed by fragmentation of the bones, and lastly followed by corrosion, which is a combination of bioerosion and corrosion, which occurs after protective soft tissue is removed from the skeleton. These biostratonomic processes destroy the vast majority of skeletal material, and any material that survives goes on to diagenesis, which are the mechanical, chemical, and biological processes that ensue following burial. Overall, the quality of preservation of a fossil is largely the result of biostratonomy and the early stages of diagenesis, which are closely related to the depositional conditions at the time of burial, and therefore can be used as proxies for paleodepositional reconstructions. Here's a table showing the biostratonomic features of different skeletal types based on the depositional conditions. Along the the right or left column is depositional energy, which ranges from high to low. And along the top is sedimentary or sedimentation rates, which ranges from very rapid to low. So for example, if you have a multi-element skeleton that's deposited in a low depositional energy with very rapid rates of sedimentation, you would expect the skeleton to be completely articulated and fairly well preserved. However, if you have the same skeleton that's deposited in a high depositional energy environment with relatively low rates of sedimentation, you would expect that skeleton to be largely disarticulated and even have the various bone pieces sorted by size. So Kidwell, 1991, defined 4 broad categories of marine fossil accumulations. First are event concentrations, which represent single depositional events and are typically contained within a single laminae or bed. Evidence of these concentrations might include erosional surfaces or infilled burrows, which typify rapid burial events such as turbidity flows or storm events. The fossils found in this type of concentration might be articulated and well preserved. However, there are a large array of scenarios that can produce these type of accumulations, and therefore they can show great variation in geometry, sedimentology, and taphonomy. Second are composite concentrations, which represent multiple depositional events and are typically contained within amalgamated beds or bed sets. Therefore, they have a more complex and lengthier history than a single depositional event, and typically are equal or greater in thickness to surrounding strata. Not all of the events may be preserved, and each event may have a unique set of characteristics, but when you combine the characteristics together, you can get the information about the depositional conditions over time. Evidence for this type of concentration may also vary with regards to thickness, sedimentology, and taphonomy. Third are condensed or hiatal concentrations, which are similar to composite concentrations. However, they're thin relative to surrounding strata due to low net rates of sedimentation. Evidence might include poorly preserved fossils, extensive biotubation, and mineral precipitation, all of which are indicative of prolonged exposures on the seafloor. And last, we have lag concentrations, which represent significant stratigraphic truncation, typically from erosion or corrosion, which uncover and concentrates usually larger skeletal pieces and sediments via selective removal. Evidence might include well-sorted coarse-grained matrix, and fossils may exhibit unimodal or bimodal preferred orientation with high degrees of fragmentation and abrasion. A whale fall involves an individual whale carcass that's deposited on the seafloor, and they are an abundant source of organic nutrients for organisms in a setting that's typically scarce in resources. And Smith and Bako 2003 documented that carcasses undergo a predictable sequence of ecological stages. Each of these stages can yield distinctive sedimentological and taphonomy characteristics. However, it's also possible that extreme conditions such as low temperature, pressure, and anoxia may actually halt or hinder biologic activity. So the first is mobile scavenger stage, where the majority of soft tissues are removed via mobile scavengers such as sharks and hagfish, as you see on the left. Evidence might include bite marks, scratches, and grooves on bone surfaces, as well as embedded or loose fossilized shark teeth. This stage may take months to years to complete. So then the next stage is the enrichment opportunist stage, which involves heterotrophic macrofaunal species such as polychaetes and crustaceans, as you see in the middle photo. Evidence might include fossil macrofauna, burrows, and borings on bone surfaces, and trace element enrichment of the sediments. And this stage also may take months to years to complete. Third is the sulfophilic stage, which involves sulfide-tolerant chemoautotrophic bacterium, which gets their nutrients from oxidation as opposed to photosynthesis. These bacterium feed on sulfide being emitted from the anaerobic decay of bone lipids and can promote in vivo precipitation, which can preserve any remaining soft parts. And then there is actually a fourth, largely theoretical stage, which is called the reef stage, and is thought to include epifaunal suspension feeders, which colonize the hard substrate of the skeleton. This can only occur once all soft tissues are completely removed, and can take up to 5 decades following death of the whale, depending on the size of the carcass, from beginning to end. The geologic setting here in Oragne County during the early to middle Miocene was very tumultuous. We had the onset of a progressive marine transgression, which resulted in rising sea levels. We also had the opening of the Los Angeles Basin during this time, which occurred over 3 tectonic phases. And we also had, of course, a major plate boundary regime shift from the ending of the Farallon plate subduction and the beginning of the evolution of the San Andreas transform fault system. This led to an array of tectonically subsiding marine depocenters along the continental margin, and all of which led to the accumulation of one of the most complete marine and terrestrial fossil-bearing sedimentary sequences known from this time period from the world over. So it's pretty cool stuff. The This is a schematic I modified from Cooper and Solon 2008, which shows the fossil-bearing strata and their depositional environments as seen in the San Joaquin Hills during the Miocene. I've highlighted the topanga formation in green, which is divided into 3 members. On the bottom, you have the lower bomber member, and then followed by the middle Los Trincos member, and then the upper Palorino member is the youngest. The Palorino member is found interfiguring with the Sedimentology breccia, as shown, which is derived of Catalina schist, and was deposited in an outer shelf to continental slope environment with periodic gravity flows. It was also deposited during the end of the transtension and the beginning of the transpression phases of the Los Angeles Basin evolution during a period of high combination space, shown all the way to the right of the figure. And here's a simplified cross-section of a transect between the San Pedro Basin to the left in the west and the Santa Ana Mountains to the right on the east. This is also of Miocene strata shown in yellow, as seen in the San Joaquin Hills. The location of the future San Joaquin Hills is highlighted in green and consisted of 3 fault blocks that were later and locally uplifted. Here, the marine strata were deposited near epicenter of what's called the Topanga formation or basin, which was located on the edge of an actively subsiding structural trough called the Capistrano embayment, shown right here. So we're talking about this area somewhere in there, which is where we believe the San Joaquin Hills and Bonita Canyon site was deposited. Bonita Canyon is located on the northern flanks of the San Joaquin Hills, which is approximately right here. To the west, we have the Catalina schist, which is the source of the San Onofre breccia, and the Santa Ana Mountains to the northeast, and the Los Angeles Basin is to the north. The inset map shows the location of the Bonita Canyon bonebeds, roughly in yellow here, on an east-facing slope of a tributary to the Bonita Canyon, which runs parallel to the 73 toll road here. The Bonita Canyon Planning Area 26 was a grading project conducted by the Keith Companies in 1997, which uncovered 3 cetacean bonebeds. From this site, over 11 articulated to semi-articulated cetacean species was removed during excavation. These consisted of large and small Mysticeti, 1 primitive Archaeoceti, and possibly 1 Odontoceti, with 1 rare or previously undocumented cetacean species. Also with the whales were found fragments of teeth from several shark species, invertebrate burrows, coprolites, and fish fragments, as well as woody plant fragments and 2 leaf impressions. Here's a section of the stratigraphic column that I modified from John Cooper's field notes taken during excavation of Bonita Canyon. Note that there's approximately a 1 meter thick section of sediment missing from this view, just so I could fit it all on the screen because it was fairly large. The lowermost sediments described at the site consisted of dark gray to gray massive tufaceous siltstones with lenses of poorly sorted sand, pebble, and conglomerates, which are typical of the Palorino member. However, the uppermost sediments were described as tan to brown, fine, of course, silty sandstone with oxidation streaks. These sediments were originally mapped as Capistrano Formation at this location. However, the Keith Companies proposed in their mitigation report that these sediments may just represent a physically different facies of Palorino or redeposition of and reworking of older Palorino sediments. Also note, because I don't show this again later, but that the section does coarsen progressively upwards, as you can see here, and even in the larger strat column, you definitely see this upward progression of coarser grains. I also want to note that the mitigation report describes isolated specimens occurring above and below the 3 bone beds, highlighted in red, where the report states that the upper isolated specimens, as well as the bone bed 1, which is the uppermost bone bed, consisted of scrappy fragments that weren't considered worthy of salvage, where the lower 2 bone beds were described as the main bone bed. So here's a map I created from different lines of evidence from the mitigation report showing a 1 meter by 1 meter grid network that was set up in the main excavation area that was called the North and South Block. The area is roughly 20 meters by 25 meters. The yellow grids contain specimens that were considered worthy of salvage, whereas the orange grids contain specimens that were considered not worthy of salvage, and then the green grids contain both, some specimens that were collected and some that weren't. I included the inset map again to show the layout of the bonebed excavation as it relates to the actual site location on the east-facing slope. So note that to the left is going up this slope of the tributary, and then to the right is down slope. There was also a second smaller excavation area that was approximately 100 meters south of this location. However, it was considered outside of the scope of recovery, so those fossils were preserved in situ, and there was no further description other than they were well preserved and articulated. So why is this study important? First, the paleoenvironmental conditions during the Rhyolithian and Louisian age of the Middle Miocene here in Orange County is disproportionately lacking. Much fewer fossils have been recovered from sediments of that age, where Bonita Canyon represents a vast majority, and some people actually estimate up to 99% of the fossils discovered for this time period. Also, little is known about the taphonomic processes and depositional conditions that can lead to deep marine fossil accumulations, where even less is known about the conditions that can lead to multiple bone beds within a single stratigraphic unit, like those found at Bonita Canyon. Very few well-preserved and articulated cetaceans have been documented here in Orange County and even the world over, where multiple cetaceans that are well-preserved and articulated found together are even more extremely rare. This represents the first comprehensive scientific analysis of the Bonita Canyon bone beds, where the primary goal was to test a hypothesis in the Keith Company's mitigation report that the main bone beds accumulated in a low-energy deep marine environment over a prolonged period of time as a result of low net rates of sedimentation. So I collected data from jacketed specimens. There were 14 total that were either opened prior to or during my study, which included a total of 27 skeletal elements, from which I collected taphonomic features on a scale of 1 to 5 for abrasion, fragmentation, remineralization, bioerosion, and the overall quality of preservation. I also recorded sedimentologic features such as the matrix color and any erosional surfaces, grading, or bioturbation that I saw. I collected 13 sediment and bone thin section samples for petrographic analysis. I collected 6 sediment samples for microfossil analysis, 18 bone and sediment samples for geochemical analysis using inductively coupled plasma emission spectroscopy, in Dr. Wood's labs. The photo shown here is of a jacket that I actually opened during the study. That was pretty exciting because I got to use a power tool. I also collected data from the field notes from the mitigation report. There were approximately 99 field illustrations from which I could determine the degree of articulation from approximately 150 skeletal elements from the field map reconstruction that I've actually set up to the left of the screen over here, where articulated to semi-articulated specimens were considered in anatomical position or near anatomical position. Associated specimens were considered not to be in anatomical position, but associated together or found together. And disarticulated specimens were isolated. For example, here's a field map that I've included here that shows both rib sections that are somewhat aligned together. And there, if you look carefully, there's actually a semi-articulated vertebral column from which I was able to collect paleocurrent data from using in situ azimuthal measurements of elongated skeletal elements such as the ribs, and from articulated components such as the vertebral column, such as shown by the red lines here. So on to the results. The mitigation report included the bonebed of origin for only 7 jacketed specimens. So I was pretty confident that I could determine the bonebed of origin using several different lines of evidence for the remaining jackets. First, I attempted to use a collection status. So recall that the report states that specimens found in bonebed 1 were not considered worthy of salvage. Therefore, you could deduce that collected specimens may have come from bonebed 2 or 3. However, the field note or the field illustrations actually have several jacketed specimens that were noted to have come from bonebed 1 and were collected. So that was an inconsistency. Second, I tried to apply the law of superposition for grids that contained overlapping specimens. However, the stratigraphic thickness between specimens was not listed anywhere in the mitigation report. Therefore, I was unable to determine if these overlapping specimens came from the same bonebed or from separate bonebeds. Then I went on to the jacket numbers, which numerically I tried to determine the bonebed of origin for specimens that had lower jacket numbers coming from, or being collected first and coming from higher in section, and jacket numbers that were higher were collected later and therefore came from lower in section. However, there was no indication of the chronology of specimen removal listed in the report. So it's entirely possible that specimens were removed from the site in sections top to bottom, moved over, or some other more chaotic fashion. And then lastly, stratigraphic correlations of John Cooper's strat column with the sedimentologic features that I observed from the 14 jackets. But there was a discrepancy between the mitigation report and the strat column's descriptions of the 3 bone beds. So this was an unreliable source. Therefore, this was largely unsuccessful due to these inconsistencies and some lacking information. So visual and petrographic analysis of the 14 jackets confirmed that 2 mean sediment types did occur at Bonita Canyon. However, the 14 open jackets, 7 of them had limited sediment above level of bone because they were previously opened, and that sediment had been discarded. And 2 jackets had very little or no sediment at all. The photo on the left, the top left, is of a smooth cross-section of a jacket containing a medium gray tophaceous siltstone. 9 out of the 14 jackets contain sand or gravel lenses, such as the one shown in this photograph, circled in red, which ranged between 0.3 and 5 centimeters thick. The photo on the top right is of a jacket containing tan to light grayish brown, moderately to poorly sorted tophaceous wacke. And you can see there's a 2 centimeter rounded gravel class that's up against this fairly poorly preserved fossil element. I lost my spot. And the bottom left photo is of a siltstone with varying degrees of iron staining, as seen in thin section. And the bottom right photo is of a poorly sorted wacke, as seen in thin section, where I've highlighted in red some examples of the angular blocky grains of tuff seen in all petrographic samples. 7 jackets contain planolites burrows, which were oblique to subparallel to the bedding surfaces. They range from 0.6 to 1.3 centimeters in diameter. They were unlined and infilled with coarser grain matrix. They were typically iron stained and all occurred below the level of bone, where 2 were slightly lenticular, suggesting some compaction. The top photo is of another smooth cross-section of a jacket containing burrows, which were below and oblique to the bone surface, which was at the top. And this yellow staining, so these are the burrows here, and then this yellow staining along that fracture may also be related to biorotubation. And the bottom photo is of 2 iron stained burrows, and note that this one truncates a couple centimeters below the level of bone, which is up here. So this actually may represent an erosional surface. Taphonomic results show that there are 2 degrees of preservation, as shown by this bar graph, where the y-axis is of the quality of preservation on a scale of 1 to 5, and the x-axis is the number of skeletal elements seen in each of the jackets. There's actually 13 here, because 1 I didn't observe any skeletal elements from. 16 elements were considered to be moderately well preserved, with values between 2 and 3. highlighted in yellow. And 11 elements were considered poorly preserved with values between 3.5 and 4.5, highlighted in blue. So the top photo is an example of a well-preserved cetacean fossil with minor fracturing. All the elements had some degree of surface flaking, cracking, or sawtooth fractures, but perpendicular fractures which cut through both bone and sediment were considered post-burial, even possibly related to removal of the specimens, and therefore were not included in the fragmentation ratings. The bottom photo is of a poorly preserved cetacean fossil, which is actually almost completely degraded. I was unable to give bioerosion and abrasion ratings to some specimens because the bone surface was completely missing. So the degree of articulation was determined for over 150 skeletal elements from the field map reconstruction seen over here to the left of the screen. 46% of the 150 were considered to be articulated and semi-articulated, where 30% were in association and only 9% were actually disarticulated. I wasn't able to determine the degree of articulation for 15% of the 150 due to the poor quality of some of the field illustrations. So no remineralization or compaction was seen in the bone material. However, I did see black coatings in 5 of the jacketed samples and 5 of the thin section samples. The top photo is showing irregular blotches on bone surfaces, which ranged between millimeter to about 1 centimeter in diameter. The bottom left photo is of a petrographic sample, of both sediment, which is at the top. This is sediment, and this is bone material, where a black dark veneer was seen on bone surfaces, shown by the red arrow, and dark spheres were seen in Haversian canals, shown by the red circles, both which ranged about 1/4 millimeter in width. And the bottom right photo are of blotches which form Lysigong rings, which are concentric circles or blotches surrounding bone elements. So here's the bone element roughly right here. So these are these blotches that form these concentric rings around up to 1 centimeter around skeletal elements. So as mutual measurements of the 99 elongate and articulated components were plotted on rose diagrams to show any preferred orientation, which can indicate paleocurrent direction. The plot A are of all measurements collected, which show a strong northeast-southwest paleocurrent direction, shown by the red arrow. Plot B are of specimens determined to have been collected from Bonebed 1 per the field maps, which also show a strong northeast-southwest paleocurrent direction. And plot C, however, are of specimens collected from Bonebed 2, which show more erratic orientations with a weak north-south paleocurrent direction. I didn't have any record of specimens coming from Bonebed 3. 6 sediment samples were analyzed under magnification for foraminifera, which can provide indications of paleodepositional depth in marine settings. However, only 1 sample contained 2 bone fragments, which were likely diatom spicules, which unfortunately don't provide any paleodepositional information. So the 18 sediment and bone samples analyzed for geochemistry, namely phosphorus and iron, where 7 samples, overall they range from 0.1 to 6.4%. And when you compare that to the value for average sale of 6, 7 samples were considered to be enriched, which suggests pore waters became saturated with respect to phosphate as the whale carcasses decomposed. And for iron, ranged from 1.73 to 7.67%. When you compare that value to the average shale, which is 1, only 2 samples were, 2 samples were considered slightly elevated over that value, which suggests that anaerobic sulfate reduction occurred in order to continue the decay of the organic matter as oxygen was depleted in the system. And however, we do consider that the black coatings are iron sulfide mineral precipitates. So recall that the sedimentation rate and depositional energy at the time of carcass deposition largely determines the degree of articulation and preservation of fossils. So for Bonita Canyon, we do see well-preserved and articulated fossils, which suggests that very rapid or relatively rapid burial did occur. However, we have varying rates of preservation and articulation to some degree, which suggests that the depositional energies and rates of sedimentation also varied somewhat for some specimens. The Planolites burrows below bone suggest that some specimens may have reached the opportunist stage, ecological stage 2. However, recall that some burrows did truncate several centimeters below bone, so those were likely infilled before deposition of the skeletons. The iron sulfide minerals likely precipitated during bacterial growth, which suggests some specimens reached the sulfophilic ecological stage 3. However, Ellison et al. 1991 also showed that mineral precipitation of this type can occur during shallow burial. And lastly, the prominent northeast-southwest paleocurrent direction corresponds with previous studies, specifically by Blair 1979, which demonstrated a southwest flow in a submarine fan depositional environment for the Topanga Formation in the San Joaquin Hills, which suggests that Bonita Canyon was also deposited on a submarine fan in depositional environment. So overall, I was able to identify 3 taphonomic categories for the fossils at Bonita Canyon, where the first were high to moderately preserved and articulated, which were buried most rapidly out of the 3 categories before significant removal of the protective soft tissues, either by decomposition or scavenging. Second are high to moderately preserved fossils, which were associated to disarticulated, which may have seen some scavenging or decomposition of the soft tissue, but were still buried relatively rapidly and owing to moderately to high degree of preservation. Third are poorly preserved and semi-articulated to disarticulated fossils, which also saw some degree of scavenging or decomposition, like possibly even transportation, and also experienced prolonged exposures on the seafloor, which caused the bones to be more degraded. So assignment of Bonita Canyon to the Palomino member is supported by 2 facts. One is the presence of a foraminifera, which was identified during excavation and is a marker for the Middle Miocene, Milesian to Luisan age sediments, and also by the angular grains of tuff seen in all petrographic samples during this study, which correlates with the descriptions that Palomino member sediments are highly tuffaceous, and the angularity of these grains also supports this with a local volcanic source for the tuff grains with simultaneous deposition with the bone beds. So I was able to construct a general depositional model for the 3 bone beds based on evidence from the mitigation report. I was unable to correlate the taphonomic and geochemical data collected during this study because I was unable to determine the bone bed of origin for the majority of the jackets. And bone bed 3 was the first to be deposited, so is the oldest of the 3 bone beds. So we know it was deposited in a massive siltstone, which indicates a low depositional environment. The bones were well preserved to articulated, which suggests high rates of sedimentation. The report also describes a accretionary silt around the fossils, which suggests mineral enrichment from the decomposition, which also supports rapid burial for this bone bed. And while I was unable to collect any paleocurrent direction from this bone bed because there's no specimens indicated from this bone bed, it likely predates development of the submarine fan, and therefore represents accumulation during quiet conditions with high rates of sedimentation in an outer shelf to continental slope environment. So bonebed 2 we know was deposited in a sandy siltstone, which coarsens upwards to sand with some gravel, which suggests relatively low depositional energies but increasing over time. And the fossils were well preserved and semi-articulated to associated, which suggests relatively rapid burial. And the scattered fossil orientations with the weak north-south paleocurrent direction suggest that deposition occurred on the medial to distal parts of a developing submarine fan, where shifting depositional conditions were the result of increasing depositional energy, which was likely connected to the subsidence of the Topanga Basin, as shown here. And then Bonebed 1 we know was deposited in a sandy pebble to conglomerate lens that was surrounded by a massive sandy silt. This is actually indicative of high energy depositional environments. And the poorly preserved and articulated to disarticulated taphonomies suggest variable rates of burial between the specimens from Bonebed 1. And the strong northeast-southwest paleocurrent direction determined for Bonebed 1 suggests that it was deposited in a more proximal setting on a northeast-southwest trending submarine fan with continued progradation, where the poor preservation suggests that high depositional energies may have been deposited as gravity flows. So here we had partially decomposed cetacean remains that were transported and possibly even redeposited as a result of these varying degrees of, or which resulted in varying degrees of articulation and preservation. So the onset of the marine transgression likely resulted in coastal upwelling along the continental margin, similar to what we see today, which may have provided a food source for various migrating cetacean species. So upon death, currents within the embayment here in Southern California would have transported the whale carcasses to near where Bonita Canyon Planning Area 26 is located today, where they sank to the seafloor. The varying biostratonomic features of the specimens within each of the bone beds indicates that deposition was not likely due to single mass die-offs. So they likely represent multiple mortality events where the cetaceans died independently. However, the mitigation report does describe some clustered skeletons in Bonebed 2 and 3 that may have been the result of mass die-off of a small pod of cetaceans. So in conclusion, Bonita Canyon actually does not represent condensed facies that was deposited over a long period of time with low net rates of sedimentation. Therefore, it was actually, it represents a composite concentration, which is initially deposited in a low energy depositional environment in continental shelf to slope during a period of high rates of sedimentation, where increased depositional energies over time were likely deposited as high energy gravity flows, which periodically swept across a prograding submarine fan environment. The high rates of sedimentation were likely the result of the high accommodation space during this time period as a result of the progressive marine transgression and the rapid subsidence related to the Los Angeles Basin evolution. So overall, this study provides insight into the Rhyolithian and Louisian age depositional conditions here in Orange County, as well as the processes that can lead to this rare class of deep marine fossil accumulations, where you have multiple well-preserved and articulated cetacean specimens in multiple bone beds within a single stratigraphic unit. And this is also the first known occurrence, as far as we could tell, of a composite concentration of cetacean remains. So further research is needed to prepare and describe the remaining jacketed specimens, especially the 7 that have recorded bone beds of origin. That would be useful. Also identifying the age, gender, and species of the individual specimens in order to try to determine the causes of death and relationships of the clustered or isolated specimens, and to determine why there's so many cetacean fossils in this one location at Bonita Canyon, which is not seen anywhere else, especially in the Palomino member in Southern California. Also, to further define the paleodepositional history of the individual bone beds and the aberrant nature of the uppermost sediments described at Bonita Canyon that were originally assigned to the Capistrano Formation. And lastly, to better place the Bonita Canyon into the overall context of the Los Angeles Basin depositional history. And that concludes my defense, so I'd like to open it up for questions.